Well, welcome everybody. This is our next installment of the um, molecular ML reading group. We're thrilled to have Ian Dunn from the Koes lab. And he'll be telling us about accelerating inference in molecular diffusion models with latent representations of protein structure. And yeah, we're really excited to have him here. Let's take it away.
Cool. Thank you. Okay, yeah, hi everybody. Thank you for being here. Yeah, so my name's Ian. I'm going to present this paper, which was accepted as a spotlight paper at the NeurIPS 2023 Generative AI in Biology workshop. And yeah, okay, so let's get into it. So I'll start a bit broad. What I'm specifically interested in, or what my research is generally focused on, is structure-based drug discovery. And so, what I mean by that is, given that we have a particular protein target, which may be associated with a disease or something, like we want to answer the question, which molecule might bind here, and might bind to this protein. And even though we can state it really simply, it's actually a quite an important question to be able to answer. You might say it's a $1 million question, but actually in this case you would be wrong, because this is a $50 billion question, which is the lifetime revenue of this compound that you're looking at. And that's not just to say that we should be measuring the sort of the value of scientific research in terms of monetary outcomes, but it is to illustrate that this is if we can answer this question well, it can have a lot of impact. So, how do we do this? How do we go from, a particular protein target to finding a molecule which might have activity for it? And there's a lot that goes on in here that makes this really hard. But, one component that makes it difficult is simply the space of potential drug-like molecules is massive. It's on the order of 10 to the 60. There's a lot of different estimates, but something like this. And for context, there's 10 to the 80 atoms in the universe. So this is an impossibly large a space of molecules to search there. And the majority of structure-based drug design as it is today proceeds through what I would describe as screening-based approaches. And that is, given that we have a protein, a particular target, and a specific ligand that we might be interested in, we want to evaluate the affinity that these two objects have for each other. Do they bind to each other in some way? And then we get some sort of score. We want to quantify this sort of affinity in some way. And I'm being really vague here, but this affinity evaluation method, could be physical experimentation, or it could be computational methods. It could be like molecular docking or physics-based simulations. But all of these methods, what they have in common is the input is like a given protein and a given ligand, and you want to evaluate if there's affinity here. Are these things going to bind to each other? And so the screening comes in where you do this evaluation for many different molecules, and you maybe can sort them by their scores, and hopefully the molecules with the best scores actually might have activity for your target. And so there are a few problems with screening-based approaches. First, the accurate methods of screening, like running physical experiments, it's really slow and expensive to do. So there's no way we can practically screen all of chemical space with physical experimentation. And then if we switch to faster methods like molecular docking, they're actually really inaccurate. So even if you do molecular docking for screening, you usually have to follow it up with extensive physical experimentation anyways. And even though they're inaccurate, you can also argue that these fast methods aren't even fast enough. If you have lots of computational resources, you might be able to dock, millions to billions of molecules into one receptor, but that's still not anywhere close to the total scale of chemical space. And finally, screening is, no matter which method you choose, physical, experimentation, or computational, it's fundamentally an expensive process because you're doing the same thing over and over. And so it's going to be inherently resource intensive. And so, what do we do? How can we get around these sort of issues of screening-based approaches? And I would argue a solution might be to simply don't do it. Just don't screen the molecules at all. Instead, we should maybe be generating molecules. And what I mean by that is that, if we can use generative models where the input to a generative model would be your particular protein target, and the output is just directly outputting the ligand that we think should bind to this protein. If we could do this well, we could just bypass the screening approach altogether. And so this is really promising because if we could do it well, we could potentially accelerate drug discovery, and we could also potentially improve drug discovery, whether it's through maybe a more efficient allocation of resources or through enabling things like rational drug design. And so this is kind of an attractive prospect if we could do it well. So the field of generative models for structure-based drug design is new and it's nascent. The first paper to do generation of molecules in 3D conditioned on the 3D structure of a binding pocket was only published in 2020. And since then we've gone through a couple of different generative modeling paradigms and we've played with different representations of molecules. And today like right now the predominant sort of methodology is diffusion models applied to point clouds or graph-based representations of structure. And this slide might be about to be outdated. I'm sure this is going to change in a year or less. But anyways, that's kind of what's going on right now. And so I would argue, I personally think diffusion models are a promising generative framework. I think they have appealing properties. But it's not to say that they've kind of like solved any like real problems. Current like state of the art models definitely have some very real issues, and they're not quite ready for practical use. But anyways, I think they warrant like additional study, and it's this promising direction of work in my opinion. Okay, so this paper is focused on diffusion models for small molecule design. So I'll start with just some basics about diffusion models. I'm going to stick with the differential equation based formulation just to make things a little bit simple, but there are several other equivalent formulations. So I'll start by, what we usually do is we define what we call a forward process. Sometimes it's called a noising process, or a forward stochastic differential equation is what we have here. And what this is a differential equation which it will, we can start at some data point, which is a sample from our data set. And if we integrate this stochastic differential equation, this SDE is designed so that in the limit as t approaches infinity, your distribution of observed values is going to approach some tractable stationary distribution, which we call our prior distribution. So in this case, it's like in the limit as t goes to infinity, the pixel intensity values all become sampled from a standard Gaussian distribution. And so the interesting thing, so going from data to noise, that's pretty easy to do. But the interesting thing about stochastic differential equations of this form is that there exists also a reverse stochastic differential equation. And this is what we call a reverse process. It goes backwards in time. From our, from this, noise distribution to presumably to data. And the only component of this stochastic differential equation that wasn't in on our forward process is this term here, which we call it's called the score function. This is the gradient of log probability of your current data point. So what this essentially is saying is, given my current data point, in what direction is probability? What direction should I move it? Under this process, and what direction does the probability increase? And so the idea is if you want to be able to access this reverse process so that you could take samples of noise from your prior distribution and turn them into data samples, all you need to be able to do is integrate this SDE. And the single piece of information we need to do that is this score function. So training diffusion models essentially boils down to approximating this score function with a neural network. And then running inference with these is once you have your approximate score function, all you can just plug in here, do integration of this reverse process, and you can recover data samples from noise. That's the goal. And this kind of gets at an important point I want to emphasize here is because what our neural network is parameterizing is sort of the drift term of this differential equation. If we want to generate a sample, we have to do multiple evaluations of this neural network in order to get a single sample. Okay, which is an important point I'll get to later. Okay, so moving on, how do we then take diffusion and apply it to molecules? And this has kind of been an interesting developing field. So before I can answer that question, I'll talk about how exactly do we define molecules. Here we're looking at just a small molecule. And obviously a molecule has atoms, but specifically these atoms, they each have a position in space. These are Cartesian coordinates. These elements can also have, or these atoms can have types, or which are like elements: carbon, nitrogen, oxygen. If we want to be really rigorous, atoms can also have formal charges assigned to them, and there can also be bond orders between pairs of atoms. I'm actually not going to be too worried about that in this talk. And the models that I'm going to present today, they just generate only the positions of the atoms and the atom types, and we try to infer the rest later. But the point I'm trying to get at here is like a molecule is actually a mixture of these distinct data types. And so now we can move forward to the question of how do we define diffusion on molecules? And so this is, I've kind of like broken it down, and this is a general strategy which works for not just small molecules, but proteins and, in other tasks as well. So what we do is the first step is we define what are the constituent data types that make up the structure, the thing that we're interested in. So as I mentioned before, in this case, it's the positions of the atoms and the atom types. And then what you do is you define independent noising processes, independent forward processes on each of those data types. So each the forward process can be defined in terms of these diffusion kernels. So this is the probability of your positions at time t given the positions at time 0. So these are noisy positions given completely like denoised clean positions. And if we define it, we usually define it this way, such that, as time goes to infinity, we push this distribution towards becoming a standard Gaussian distribution. And so this happens independently for both positions and atom types here. And then once you define this like independent forward processes, what you do is learn a joint reverse process, right? And so we train one neural network to approximate the score functions of all of these constituent processes. So this is like our loss function here would be, there's two terms in this loss function. It's the score function on atom types and the score function on positions. And then, we have, here's like, these would be two different outputs of like the same neural network generally. And you're trying to like minimize these score functions jointly at the same time in the expectation over all pairs of positions and atom types that you would observe. So this is the general procedure for doing diffusion on molecules. Here's an example of what it would look like. So in this forward process, you see the positions of these atoms kind of collapsing to our prior, which is a Gaussian distribution, centered at the origin. And then in the reverse process here, you can also note that the atom types also become random as you go in this forward process. And in this reverse process, what the model has to do is it has to predict updates for every atom. So the positions and the atom types have to be updated for every atom. And so, this whole sort of diffusion process becomes like a node-wise prediction task. You have to make a prediction for every atom. In order to do this, under the hood, we're using graph neural networks. So every atom becomes a node in a graph. Okay. So this is, generally how diffusion works on molecular structure, how we can do it. And if we want to take this, a step further, what we're really interested in, what I'm interested in, is being able to do diffusion inside of a protein binding pocket. So we want to, given the 3D structure of a binding pocket, like, generate a molecule inside of it, which is what we're looking at here. I'm not the first person to do this. That's not what this paper is presenting, but so like generally other people have done this, and this is generally how it works. Is you have a diffusion model, which it operates on like your ligand atoms and refines their positions and features in 3D space. And then while this is happening, the structure of the protein binding pocket is provided as context for this diffusion process to take place. How exactly do we, allow this like binding pocket to communicate with the ligand? We use heterogeneous graphs as our sort of representation here, right? So we convert this ligand receptor complex into a graph, which is what I'm showing you here. I've taken just the protein ligand complex, just a protein binding pocket, and I've turned every atom into a node and connected these nodes. You can see if you look closely, there's actually edges that occur between ligand atoms and receptor atoms. And so once we have this structure, if we apply this as like input for a graph neural network, we allow the structure to communicate in some sort of learned fashion with the ligand. And this pocket becomes context for this diffusion process. And so this is generally how we do diffusion of ligands inside of a protein binding pocket. Okay, so I've like gone over quite a bit of background. I've jumped back to this title slide because now I'm going to start focusing on it, like precisely like what we're doing in this paper that's actually new and novel. This issue I kind of notice is that these protein ligand diffusion models, they tend to exhibit relatively slow inference times. Specifically, you know, like we know that slower inference times are inherent to diffusion models. I've mentioned this. You have to do multiple neural network evaluations in series in order to generate a single sample. So that's like an inherent limitation of diffusion models. And then when we take these models and we apply them to graph-based representations of structure, we have this problem because graph neural networks don't scale very well with the size of your system, right? And particularly when we add a protein into our structure now, it dramatically increases the size of our graphs, and we get this big slowdown. And so this kind of like presents what I call this like protein representation problem, which I've seen in the literature or saw, which is, some people, there's two things you can do. You can represent your protein binding pocket in two ways. Some people will do an all atom based representation of the binding pocket. So there's a node in your graph for every atom that's in the binding pocket. And some people will do an alpha carbon only based representation, right? So there's one node for every residue that's in the binding pocket. And this is also the alpha carbon based representation is also more common for, if you're doing whole protein design tasks. But there's kind of like this trade-off here where, if you have all atom, you're talking, on an order of magnitude more nodes in your graph. It's much more expensive to do, but it'll give you better performance. It's kind of the situation. But so some people will use alpha carbon based representations. Some models use alpha carbon based representations just to make like training and inference more tractable, because it is much simpler, but you lose out on a lot of essential information, particularly for like binding events. The orientation of side chains is like really important information to have there. And so, you can look in a few different examples and see like when we only use alpha carbon based representations, we get some serious degradation in the performance of models. But then the odd representations are being really expensive. And so there's this trade-off here. And, in this paper, we try to like ask the question, can we have both, or maybe like the best of both worlds? And what I mean by that is like, can we ameliorate this trade-off? Is there a way to obtain a protein representation that's both small, it's fast and compressed like the alpha carbon based representations, but it's informative and is performant, enables us to generate a high quality ligands. So coming back here to sort of what the typical setup for receptor conditioned diffusion looks like, we have our binding pocket. It acts as conditional information. And what we do in this paper is we introduce Keypoint diffusion is what I call it. And specifically, there's this component here, which we call a receptor encoder. The input to this receptor encoder is the all atom binding pocket, and the output is what I call a keypoint representation. This is a small fixed size point cloud, or you can think they end up being nodes in our graph. But there's a fixed number, which the number is a hyperparameter of the model, and they're produced by this receptor encoder, but the receptor encoder is basically just a neural network architecture. And so it's continuously differentiable, and we can actually learn whatever this key point representation is. These key points, they have positions in space, as well as they have invariant features and some equivariant like vector features attached to them. And so then this learned key point representation can then take the place of this binding pocket and become the conditional information for our diffusion model. And because it's all continuously differentiable, we can train end to end, and we can learn the representation of the binding pocket that is most useful for this generative task. Specifically, like what does this look like? I can show you this is an example. I've shown you this video a few times, but I neglected to give you kind of the details of what's going on. So this is a molecule being generated inside of a binding pocket, but it's actually being done by our Keypoint diffusion model. And so what you can take away from this view is that generated ligands seem to be conditioned on the binding pocket. They fit inside of it and have some sort of complementarity for it. But in practice, what's actually going on, or like what the model is actually seeing, is this. The model only ever sees this key point representation, right? So this is 40 points, these blue dots here, they're like 40 points. They have positions in space. And this is the only representation of the binding pocket that the model ever actually sees. But despite that, it's able to actually design molecules for the binding pocket. And the advantage here is, if we have this smaller representation, we can get faster inference just due to the reduced size of the graph. But because this representation can be learned, potentially we can get more something that's more informative and that allows us to produce high quality ligands despite its small size. Okay. I'll jump into some of the details of like how does this receptor encoder actually work under the hood. And most of this receptor encoder can boils down to controlling which edges we're doing graph convolutions on or message passing on at a time. So the first step here is the input is protein, a binding pocket, and as a graph, and we do message passing operations, graph convolutions, only along edges that are between receptor atoms. And this allows receptor atoms to obtain some sort of learned representation of their local environment. And then after this step, what we do is we introduce these key point nodes to the graph. Initially, these key point nodes have no features associated with them. They don't have positions in space or anything. But we're going to assign them positions in space as a first step. And the way we do that is completely learned. So the position of one key point is going to be a weighted sum over the positions of all of the protein atoms. And these weights are learned. It's a graph attention mechanism. So the model decides on its own to place key points, somewhere in this binding pocket or around specific sets of protein atoms. And then once these key points have positions in space, what we do is we rewire this graph. So now each key point has incoming edges, but only from receptor atoms, which are close by, nearby. And then after we do this rewiring, we can do message passing again, but only along these edges between receptor atoms and key point atoms. And this way we're kind of pushing information from the receptor into the key points. But in a way, because of this sort of localized wiring scheme, each key point only ends up receiving information that is specific to its local area in space. And so that's how these key points end up sort of summarizing local structure. Okay. There's one extra layer here. We have this extra loss function that ended up being quite important for getting this model to work. And so the motivation here is to try to sort of enforce the key points that have some physical meaning. And the intuition is that if these key points should have all of the information required to design a small molecule for a particular binding pocket, the intuition is that maybe these key points should be able to describe the types of interactions that might be occurring between the protein and the ligand. In order to do that, they should probably be located along this, whatever the actual true interface is where this ligand would be coming into contact with the binding pocket. And so at training time, we can actually compute a set of what I call binding interface points. These are points where the protein and the ligand come close into contact with each other. And for a true ligand that is actually in the data set and known to bind to this protein. And then we have this optimal transport loss function here is a loss function applied to the predicted positions of the key points. In this loss function, I'll skip over the sort of mathematical detail here unless anybody wants more information. But the effect of this loss function is that it is minimized only when these key points, these green points, align collectively with all of the binding interface points, right? So if there's up here, if there's a binding interface point, this is a location where the protein is actually coming into contact with the ligand, but there's no key point placed anywhere near it, the loss function is going to go up. So what happens when we apply this loss function, it means that the receptor encoder, which takes as input just the binding pocket without a ligand, has to figure out where would a ligand actually be sitting in this binding pocket. And we find that that's a crucial sort of inductive bias or learning signal in order for this receptor encoder to learn useful representations of the binding pocket. Okay, so I'll start to walk through, the experiments we did to evaluate the performance of this model. We trained on the binding MOAD data set, which is just pretty much every structure in the protein data bank, which has a small molecule in it with some filtering rules applied. So it's about 40,000 protein ligand complexes, crystal structures. And for baseline models, we train two different baselines, and the difference between them is the representation of the binding pocket. So otherwise, the exact same model as our Keypoint model, but one uses an alpha carbon-based representation of the binding pocket, and one uses an all-atom-based representation of the binding pocket. And finally, we run some experiments with the expressivity of the graph neural networks that we use under the hood. I can talk about this in a bit more detail later, but this ended up being important. We had started with EGNN, and it would turn out geometric vector perceptrons ended up being a better choice. Okay, in order to evaluate, I'll talk about what do we use to evaluate the performance of these models. So we're mostly focused on the complementarity of the ligand for the receptor. How well does this ligand fit in the binding pocket? So we do this with two metrics. The first thing we do is we would generate ligands inside of a binding pocket. Then we would do a force field minimization of the ligand inside the binding pocket. So we keep the binding pocket fixed, but at least the energy of the binding pocket is included in the force field. And so once we do this minimization, we measure the RMSD of the structural rearrangement that the ligand had as a result of this minimization. And the idea here is if upon force field minimization, there's a really large structural rearrangement, and the ligand probably wasn't really fitting in the pocket very well, or maybe there was like two positive charges on top of each other, or something like that. So if this RMSD is lower, we can say the model is doing a better job of designing ligands for this particular binding pocket. And the second thing we do is we just take the AutoDock Mina, which is traditional molecular docking software, and we compute the value of its scoring function, which is designed to approximate like binding affinity. And so we use that as a metric to evaluate how is their complementarity for the binding pocket. And then finally, the last thing we do is, the whole kind of idea here is maybe we can get faster inference time out of this. And so to evaluate inference time, we generate 100 ligands for one binding pocket. And then we do this for all models. We just run on the same GPU, same binding pockets. And then all of these evaluations are done on a validation set of protein ligand complexes that are held out at training time. Okay, so here are the first set of results here. It's there's three graphs. I'll walk you through what's going on. These first two graphs, these are the cumulative density functions for the two metrics that I mentioned. So this one is the RMSD from force field minimization, and this one is Vena scores. So these are so basically, here if you look at this, it's this value says I don't know, roughly for this model, roughly 40% of the generated ligands had an RMSD less than 2. All you really need to know here for both of these graphs is that a curve that is higher up is better, giving better performance. And we include these metrics computed on the validation set, the ground shape ligands as well. And so you can see the kind of upper bounding the performance of these models. And then this final graph here is just the sampling time, the time required to sample. It's the time required to sample 100 ligands divided it by 100. Anyways, okay, so and there's four different lines of validation set. There's three different models here corresponding to the three different representations of a protein binding pocket. So the orange line is an alpha carbon-based representation. The pink line is just our Keypoint model, which we're presenting, and then the green line is the all-atom representation. And so what you can see is the first thing we notice is this alpha carbon-based representation gives us the worst performance, which is consistent with previous works and makes sense intuitively. And correspondingly, the all-atom-based representation gives the best performance. And our Keypoint model actually lies somewhere in between for both of these metrics. And if you look at the numbers precisely, you could say it's.
Can I ask a quick clarifying question here?
Yeah.
You said there was a hyperparameter, which was the number of key points that you could use in this. So what did you use in this context?
For all of these graphs, it's 40 key points.
And these are set per receptor, and even though they're differentiable, you're not learning them in the context of the ligands themselves. Is that right?
Yeah, so it's a deterministic function for like one given binding pocket. There's like one set of key points that's produced.
Got it. Okay, thank you.
Yep. Um, Yeah, okay. So what we can say is, this key point model, the quality of ligands we produce is definitely better than using this alpha carbon based representation. It's a bit closer to achieving the performance of the model which uses an all atom representation of the protein, but it does this while using an order of magnitude less nodes in the graph to represent the protein structure. And this is reflected here in the sampling time. So the sampling time for our key point model is like 3 times faster than the sampling time for this all atom based representation. So it's nice. We were able to sort of like actually get this sort of trade off. We have this representation, which is indeed much smaller and easier to work with than the full all atom representation, but we get performance that is close to it and definitely better than the naive approach of just using only residues to represent your binding pocket. Okay, and then so this is the same graphs that I just showed you. I've just added 3 lines to these graphs. So there's 3 dotted lines that have been added, and the difference here is a model that's trained with 2 different graph neural network architectures, so GVP and EGNN. So EGNN is less expressive than GVP is the situation. And it is like a theoretically less powerful graph neural network architecture. So if we look at the first thing we can do is we can compare the model which uses key points and the EGNN architecture to the model uses key points, or the model that uses the alpha carbon based representation and the EGNN architecture. And so both of these 2 lines, they appear right here on the bottom here. They're pretty much on top of each other. And so what we can say from this is that like if you use the EGNN architecture, you're actually completely incapable of learning a representation of the protein pocket, which is any more informative than just an alpha carbon based representation, which is interesting. But then, if we, then go ahead and compare, the alpha carbon to the key point representation while using like GVP, we get actually this like really big jump, right? This GVP based model is actually able to, learn some sort of compressed representation of the binding pocket. And so this is really interesting, and it kind of makes sense. It's like EGNN, it uses it has like equivariant information about structure, but it only has one equivariant feature for every node, which is just the position of that node in space. And these key points, they have to actually like summarize structure. They represent, the position or orientation or composition of multiple nodes around it. And having only one equivariant feature, EGNN fundamentally can't do that. But GVP is basically the same thing as EGNN, but you can have the arbitrary number of vector features on every node. And so that's why we see it can actually like tackle this task. And so the expressivity, if you're designing structure-based like graph neural network models, the expressivity you're using is really kind of a key concern, which isn't a completely new idea, I guess. Yeah. Okay, so that's pretty much all the results. I'll wrap up with some conclusions here. So we show that our receptor encoder can actually learn compressed and informative representations of molecular structure. When we combine this receptor encoder with the diffusion model, this receptor encoding can like substantially accelerate inference without dramatically impacting performance. And then we can show that these EGNN, these simple architectures, actually like struggle. Like generally, they will struggle on any task where a single node has to summarize structure in any way. If the structure is any more complicated than a single point mass. So, for example, you probably shouldn't be using EGNN for residue clouds, for example. And then kind of like the spiciest take that I have to offer, and maybe the most practical advice coming out of this paper, is I think there's really like no reason to be using EGNN. And I'm open to like debates on this, but if you look at the data here, like GVP is strictly like theoretically more expressive and more powerful, but then and there's not even a trade-off in terms of like the computational demands required by GVP. Like actually my GVP implementation was faster than my EGNN implementation. So I think there's kind of, if you're choosing, like a lot of the predominant if you look in the literature, there's a lot of papers putting out like they use EGNM. Maybe they use it because it's really simple, but GDP is also like really quite simple and strictly better. So, anyways, some advice or my 2 cents at least. Okay, moving forward, some like future work that could be done on this. I think that it would be nice if this receptor encoder could be kind of pre-trained or not trained end to end with the downstream task. This would right now, because we train this receptor encoder end to end with the diffusion model, we don't get any sort of like computational savings at training time. But what we could, if we could sort of maybe like only train with the optimal transport loss at training time, that'd be nice. Also, as you asked earlier, Matt, this the generation of this key point representation is deterministic. For one binding pocket, you get one key point representation, but you could imagine, or like we know for sure, a binding pocket can have multiple binding modes. And so it would be nice to be able to sample multiple potential like key point representations given just one binding pocket. And I think that would be a nice piece of work. Yeah. Okay. That is all of the slides that I have. So I just want to say thank you to my research group for being super supportive, and thank you all for listening and being here. And I'd be happy to talk to you about any questions you have.
Awesome. Thank you.
Thanks.
Let's open it up to questions.
I have a question. First of all, a great talk, Ian. And my question is regarding the key points. Do you guys have an idea on what information is being lost when we're converting the all atom representations into key points? And what exactly is making the key points implementation faster, I guess? Like chemically, what are we losing?
Okay. So there's a few different questions. The second one is very simple. Like why is it faster? It's faster just because if you the way we're defining binding pockets, we end up with having like on the order of several hundred atoms in a binding pocket. So you have a graph with several hundred nodes in it. And our key point representation only has like 40 nodes in it. So it's strictly like cheaper to run these smaller graphs through graph neural networks. Chemically, what are we losing is a much more complicated question to answer, and probably like warrants some investigation. I guess my guess would be like what we're losing is, and maybe I think that I can tell you that there are instances in which the representation struggles to the key points struggle to summarize structure, which are maybe results of design choices for the architecture. One example I can give you is I found an example in our data set of a perfectly symmetrical dimer of two sort of proteins forming a pocket, and there's a ligand in between them, and so it's kind of completely symmetrical. And the placement of the key points is based on this attention mechanism, and because the protein was symmetric, the weights, the importance weights for placing a key point were it had two equivalent importance weights on either end of the protein. And so what happened is all of the key points ended up sitting like directly in a straight line in the middle of this like complex. And so in that case, you can't actually like summarize, you really can't, it's failing to actually summarize structure. And that's kind of a result of the architecture choice. But anyways, yeah, I guess, but it's certainly a good question. It doesn't I guess that fully doesn't answer your question like chemically, like what information are we missing? I guess we don't really know because it's kind of a black box in terms of just we're learning this representation end to end with the task.
Great, thank you.
More questions?
I have a question. For like chemical feasibility, do you place any constraints on like the generated molecule? For example, bond length?
No, we don't. And in theory, if the bond lengths would be perfect. The bond lengths would match the bond lengths in the training distribution if the model had fit the training distribution perfectly. And you're kind of getting at another issue of like, are these molecules realistic in any sense? And we do know that these diffusion models that operate at the atom level produce molecules which are not synthetically accessible, and are often like physically unstable. Like you wouldn't be able to actually synthesize them in a lab or and store them. And yeah, we don't have any like sort of extra constraints applied towards that. The constraint is just like, can you reconstruct the clean molecule that we see in the training data set? But that is certainly an area of work that I'm sure people are working on.
Maybe just to piggyback on that, there are like synthetic accessibility scores. And I know people have benchmarked generative model for chemicals against these, like the guacamole benchmark set, for instance, and then also for diversity. So I'm curious on if you're giving up if you're potentially overfitting by sort of sacrificing synthetic accessibility and diversity with your methods or not.
Yeah, in the I don't have it in this presentation, but in the paper, in the appendices, like we report these metrics. And I think that both the synthetic accessibility and the diversity is not different, or it's not substantially different than any of the like baseline models or like existing diffusion models. So this particular architecture choice doesn't actually like have any obvious impacts in terms of the quality of, if you just evaluate ligand only metrics, like what is the validity of the ligand, what is the synthetic accessibility of the ligand, it doesn't actually look that different than the sort of the other existing diffusion models which don't do this key point situation.
Thanks. More questions?
I have another question regarding the baseline model. So from my understanding, you guys compared the key point model to your C alpha model and as well as the all atom model. Did you compare this to other diffusion models using a key point representation?
Yeah, it's a good question. The answer is no. That was like the plan for this paper. But because a lot of our, sort of the claim of the paper is like, oh, we can get faster inference time. It turns out comparing inference time between two models is kind of a difficult thing to do. And because there can be a lot of, if you have different implementations, like the difference in inference times could just be the GPUs that you're running on, or the efficiency of your neural network implementation, or things like that. And so I when I initially ran this paper, I was comparing against, or when I was doing this work, I initially compared against like diff SBDD, and so that was like one of the first two papers to do receptor conditioned ligand generation with diffusion models. And I had like my inference time was like 10 times faster. But the 10 times speed up wasn't actually due to the key points. It was mostly due to my code being more efficient. So anyways, that's why we ended up like I did this whole like become my own baseline situation so that I could ensure that there's a sort of like fair comparison. Because functionally, like what we're doing is the exact same thing. It's like this model here, like this all atom and alpha carbon is functionally equivalent to the diff SBDD model. But yeah, but you can actually, you can compare the you can compare the metrics here that we have in terms of like VINA scores and the synthetic accessibility, like QED, like molecule validity. Most of it looks almost the same as those existing models.
So within the diffusion model architecture, there's a number of hyperparameters that can control the inference, like for instance, the number of diffusion steps that you run. These would impact both the sampling quality and the runtime. So I'm curious on sort of how you would situate these performance improvements relative to sort of more generic types of tuning for the model architecture.
Sorry, could you say
So
that
like,
again?
for instance, you're doing, I don't know how many steps you're doing in your diffusion, but let's say you did half of that, you would get, it would run twice as fast and you'd have probably worse samples. So like, is that a better, I mean, obviously this is orthogonal to that set, but just contextualizing the level of performance that you're getting relative to these other types of tweaks that one could do.
Totally. There's a lot of sort of, and this is like a sort of a rich area of work that's ongoing to sort of like get around or ameliorate this issue with the number of time steps you need to sample. I guess what I would say is, you know, if you would probably want to reduce your number of time steps as much as you can without, like, you know, such that in my experience, there's this sort of like cliff. Like there's a certain point, a certain number of time steps where increasing the time steps more doesn't actually improve your performance anymore. That's probably what you want to use for like your minimum number of time steps. You know, if you're already sitting there, then this method might be useful as like an additional way to you know, accelerate inference. Yeah. I think that this method, you know, but it isn't sort of like it's not really like a free lunch, you know, there's definitely like a trade-off here in terms of there's some hit you take the ligand quality. You know, you have to, I guess, like be wary of that if this was like a design choice. But, you know, if you're in a situation where you wanted to scale inference and do a lot of inference, generate a lot of ligands, then this might be, you know, it might be acceptable to take some hit in sort of like the ligand quality just to be able to scale inference more. Yeah.
Thanks. More questions?
Thanks for the great talk. I had a question about the number of ligand atoms that you chose to generate. So I think you generated like 100 ligands per pocket. Correct me if I'm wrong, but was there like a range of ligand atoms that you sampled from?
Yeah. So you have to, you know, a feature of these models is that you have to predefine like the number of atoms that are going to be in your molecule. And so what I did for this, like for this evaluation here, we were only using like protein ligand complexes like from like a validation set. So we had like a corresponding, for every binding pocket, we have a corresponding like ground truth ligand. What we did is we just used like the same number of atoms that were actually in the ground truth ligand as a first pass. So they did end up, you know, they are indeed different sizes. And the set of sizes we observe is, you know, equivalent to the distribution of ligand sizes that are in this validation set.
So I guess one way you could think of these key points is in the interaction graph between the ligand atoms and the receptor atoms. You know, you could think of like the graph as being like a matrix. This is in some sense a you know, like a factor graph, or there's some way of reducing those, you know, kind of latent representations. There's many other contexts in which you have kind of a factorization or a way of reducing a set of interacting things through a lower dimensional representation. Obviously, you have a very physical representation of these key points as being points in space, so they've got, you know, kind of location and everything. But, you know, there's other ways you could think about kind of having that reduced interaction through, like, say, just straight up matrix factorization. There's some really old methods on like interaction fields. So you could think of doing it in like, you know, a reciprocal space. You could have sort of a basis and sort of like what that interaction field might be. So I'm curious if if you came up in sort of thinking about sort of more mathematically what these key points are, and if maybe constraining them to being very real points in space, maybe that you get a real interpretation of what's going on, but maybe you give up some degrees of flexibility in terms of their predictive accuracy or representation ability.
Sure. Yeah. It's a good point. one thing you can do with these key points is, you can think of these key points as like a coarse-grained representation. And, if you coarse-grain any molecular structure in like the traditional sense, like you have to have some sort of like mapping or assignment of like your original atoms to like your coarse-grained atoms. And we kind of can like recapitulate what that mapping is by inspecting just like the attention weights that assign the placement of the atoms. And that would be like nice to look at in terms of, are there any sort of I've been meaning to do this, I just haven't had time to do it, but it would be really great to look at, is there any correlation between like, do specific key points end up being co-located near specific types of functional groups, specific types of residues. Anecdotally, these key points often end up like sitting like, I can think of an example of, there's a side chain that has an aromatic ring, and the key point ends up getting placed like literally in like the middle of the ring. So like, okay, maybe that's significant in some way. But yeah, definitely there's some sort of like interpretation techniques that could be applied. People have done it. I remember there was I think it was a paper from Charlotte Dean's lab called like Point VS, where they had a graph-based neural network for predicting binding affinity, and they just had an attention layer in there, and they tried to see, like, given the attention matrix between, like, where does this ligand seem to be interacting with the protein, can we like elucidate pharmacophores, and do those pharmacophores have like some sort of like physical interpretation? And I think they had some reasonable success. It was some pretty interesting work.
More questions?
I've got a question. So one of the really cool things about like this diffusion framework is the ability to kind of guide and sample from desired pockets of your distribution. So I was wondering, do you have any sort of like plans for guiding your diffusion process in certain directions, or how might you do that, or what interesting properties might you guide towards?
Mm-hmm. Yeah, totally. I mean, I think that's like, at the beginning of this presentation, I said, like, I think diffusion models are this like promising framework. And I think this notion of guidance is one of the really unique components. Like you said, I agree with you. I've seen people do guidance in terms of this like classifier directed guidance where you can like guide towards sort of any sort of desired property you want. So that could be, I don't know, synthetic accessibility, I don't know, lipophilicity, things like that. It could be done. There's also potentially connections with physics here. So you could there could be guidance towards low energy structures, for example. I think those are promising directions. There was some work, I think it was called like discrete guided diffusion for like antibody design. It was presented at NeurIPS from Prescient. Samuel Stanton was one of the authors, and they had this a guidance term, which was a Kale divergence, on basically saying like we want to make some sort of like we don't want to perturb this and get an antibody sequence that is too far away from some like reference sequence, and so like they don't want to make these like really weird, like out of distribution. They want to be a little bit out of the distribution, but not totally out of distribution anyways. And yeah, I thought that was really interesting too. Yeah. So yeah, I think there's a lot to play with. Like technically, I mean, in terms of like classifier guidance, like if you have any function that can predict some property as a function of like your model, and then you can back propagate gradients, and you can like, in theory, probably like push towards this sort of desired property, which is a powerful component of sort of the generative view of like designing molecules or drugs or whatever.
So in your training set, you're using binding MOAD, which is, I think, a reasonable place to be. But there's a lot more pockets on proteins in other other places that don't have ligands in them. And so I'm curious if you could do like an autoencoder type of approach with this key point, where you try to generate back the atoms of the pocket from the key points as sort of a sort of self-consistency type of constraint to know that you haven't like lost too much information in the key points. And I guess maybe a more general question is that predicting back, would that be a measure of sort of how much information you've lost in the actual, in terms of representations?
Yeah, I think it's a good idea, and it was something I kind of I thought about for a while. Was if this coarse-grained representation would be nice if it was invertible. In terms of it from a training perspective, it would be like, this sort of autoencoder style reconstruction loss, some sort of measurement of how much information is lost in this like latent space. The other angle there is, if you could do a joint diffusion over the coarse-grained representation and the ligand at the same time would be nice. So maybe like allowing the protein to be flexible in some way. The primary challenge is precisely like, how do you go from some coarse-grained representation back out to the original atom structure, which it works if you have a very like manually well-defined coarse-grained representation with an invertible mapping, but we don't we don't design to have an invertible mapping, and so you could train a model to do the mapping, and like maybe it would work, but yeah, the logistics, how you would come up with an architecture to do that, are it would be a project in and of itself, I guess. So that's why I left it out.
Totally fair.
Yeah.
Any other questions? Okay, well, we're just about at the hour.
All right.
Thank you so much for coming. It was really interesting work, and I really appreciate you talking us through it. It seems like it's opening up a lot of really interesting questions and opportunities to go forward.
Yeah, thank you all for being here. I appreciate the invite, and it was a pleasure speaking to you.
Okay, see you next
Sounds
time.
good. All right, see you guys.